Okay, good morning, everyone. It's really fantastic to see so many of you here, and thank you, everyone who's made the long trip down to Exeter. This is our beautiful Streatham campus. I should caveat, it's not always this sunny in Exeter, so thanks for bringing the sunshine. Just to note, if you scan that QR code, you'll get into a sort of interactive website where you can see the agenda and do things like vote for poster prizes, etc., during the breaks. So I'm here just to really welcome you all and set the scene. And obviously, this is a really exciting time in human biology and understanding disease. We've made absolutely tremendous progress in identifying genetic risk variants that are associated with diseases. The key challenge now is obviously to really improve understanding of the functional consequences of those genetic risk variants so that we can start to learn more about mechanisms, find new drug targets, obviously develop new treatments, and work in the area of developing biomarkers and ways of stratifying patients so that we can improve their clinical outcomes. So we're obviously the UK Human Functional Genomics Initiative, and the term functional genomics, as everyone here knows, means lots of different things to lots of different people. So we're very interested in studying molecular QTLs. So this is looking at how genetic variation impacts upon measures of gene function and gene expression. And we're doing that across tissues and different cell types. And we're looking at the effects of genetic variants on expression, splicing, epigenetic regulation, and actual levels of protein. You'll hear a lot today about how technologies have enabled us to really start engineering perturbations to the genome, and obviously that then allows us to test the functional consequences of genetic variants, not only looking at what those do in terms of ablating the absolute expression of the gene, but also fine-tuning levels of gene expression, affecting things like splicing and epigenetic modifications. And then the other thing that our initiative is really aiming to do is to generate really high-quality multi-omic functional annotations that allow us to then start putting together effects of genetic variation on these different regulatory signatures. And this obviously allows us to generate hypotheses about function that can be experimentally tested using those perturbation models, some of the high-end functional readouts, and obviously this all has to take place using disease-relevant models. So these are all things that our initiative is really interested in. I'm not talking about my research today, but I have to put up one research slide because I'm here anyway. But this just highlights the power of how new technologies are enabling us to explore these types of functions. So this is using long-read RNA sequencing that's allowing us to generate novel sequence and transcript maps. And what we're finding is that there are lots and lots of novel coding regions of the genome that aren't in existing annotations. And obviously this has real implications for some of the genetic studies that are taking place. It's giving us refined annotations for doing things like whole genome sequencing analyses. So what will our initiative do? Well, our goal is quite simple. It's to catalyze discoveries into the mechanistic underpinnings of health and disease. We want to identify causal variants, model their impact in disease-relevant cell types, and do that in relevant cell types and at key stages of development. You'll hear a lot today, I think, about some of the novel technologies that are really allowing us to do this type of work. So you'll hear about advances in sequencing, particularly around long-read sequencing, our ability to characterize epigenomic variation, look at transcriptional variation, and do that at really amazing resolution, looking at individual cells in the context of spatial profiling in pieces of tissue. You'll hear from Nicola and others about some of the tools and techniques that we have that enable us to actually perturb the function of genes in relevant models and using really high content multimodal readouts. One of our goals is to really interact with some of the amazing genetic epidemiology work that's taking place within the UK. I think we really lead the world in the context of some of our genetically informative cohorts, so cohorts such as UK Biobank, the 100,000 Genomes Project. And a real long-term aim of our initiative is to make sure that the functional data that's generated as part of the work that we're doing feeds directly into those cohorts. And obviously this will involve developing expertise and capacity in data science and AI. So just a few words on the structure of the initiative. This is how we've been set up. And you'll hear today and see posters from the different clusters that are focused on different areas of research using different technologies in different contexts of different disease outcomes. Just to say that we really want to grow. So this is really our first symposium. It's amazing to see how many people are here today. So there's obviously real interest in this. But we want to encourage additional members to join this group. You can do that simply by signing up on our website. We'd love to get more funding that would enable us to expand the number of areas that we're focusing on. We're already talking about building new clusters into new areas, and increasingly we're looking to partner not just within groups in the UK but also internationally. And we have a number of partnerships already set up, and you'll hear from Sarah later about how the work of the MRC National Mouse Genetics Network can really interface with the type of work that we're doing. So this is our structure. So the initiative is coordinated from here in Exeter. We're also developing the Data Coordination Centre, and at the moment we have 4 clusters. As I said, there are posters on each of those clusters. There's the molecular mechanisms cluster that's based up in Edinburgh, that's using human tissue, genetics and AI to explore the molecular mechanisms of a whole range of diseases. There's a cluster based at Imperial led by Matt Child, and he will give an introduction to the work that's taking place within his cluster later today. There's a musculoskeletal disease cluster in Oxford led by Dom Furness, and then a cluster at King's College London that's really focused on developing infrastructure for brain organoid models to explore genetic variants associated with neurodevelopmental disorders. And then finally, we have the Functional Genomics Screening Lab, which is based at the Milner Institute in Cambridge, and Nicola will give an overview of that setup and how people in this community can interface with them and make use of those facilities and infrastructure. Just a word on the Data Coordination Centre, because in a way this is really the centre of what we're hoping to do. And the aim here is to develop a really scalable data sharing platform. And the whole initiative is really set up from the outset with an ethos on open and reproducible science. We really want to make sure that all the experimental data that's generated in the various labs that will be working across the initiative generate relevant metadata that can really make sure that the data itself is sustainable and has a legacy into the future. And if you're interested, all the protocols that are being developed as part of the initiative are already available or starting to be released on our protocols.io workspace. So please check that out if you're interested in any of our experimental pipelines. And as I said, we will be partnering with EBI and Biofair, who are here today, just to make sure that we have a real flow through of data from the initial labs that are generating it right through into a ecosystem that can be utilized by the wider research community. So how can you get involved? Well, please, as I said, sign up, become an associate member of the initiative. We will soon be launching a series of seminars and training events. And obviously this will, we hope, become an annual symposium. We will rotate this around the UK, and so look out for our venue for next year. Please do collaborate with the clusters and the Functional Genomics Screening Lab. Please collaborate with the Data Coordination Centre. We're not just there for people directly funded by the initiative. We hope that this will become a platform that can set data standards and curate data and workflows that are relevant to the wider community. If you're an ECR, we have a new but thriving ECR network. We would welcome members from anyone working in functional genomics to join the ECR network. And please do come and talk to us if you've got ideas about areas that we should expand in and areas that we should be investing in the future. And finally, one way you can get involved is to apply for some of our collaboration funding that we have available. Just a preface to a webinar that will be held in early July, and actually the details of this scheme will be on our website directly after the symposium, so check that out probably early next week. Just a few words on that. So this will be open to anyone based in a UK academic institution. We really welcome applications from ECRs, and there'll be two streams of funding. So the first is really to foster academic projects. Collaborative is always really good, and these will be ranging from £15,000 to £100,000. And then stream two is really tailored at forming partnerships with non-academic and industrial collaborators. And again, the range of awards will be the same, but the idea here is that the industrial partners also put in some match funding into those projects. So if you have any ideas, and hopefully today is a really good platform for networking and making contacts to work with on some of these projects. And just a few words, obviously the project should align with the broader goals of the initiative. They're really there to sort of strengthen collaborations between the initiative and the awardees. We really would hope that you would agree to sign up to our ethos of open and reproducible research. The application form itself will be quite short and very light touch. And then just a final thing, these are really the people who've actually put today together in many ways. This is our coordination hub. Star, I don't know, Star, do you want to stand up and wave? Probably not, but anyway, I'll make you do that anyway. So Star and Paul, I don't know where Paul is. Where's Paul? Paul's over at Reed Hall sorting things out for later, but really huge thanks to both Paul and Star because they've put in a huge amount of work to make this happen. Ben, I don't know where Ben is. Ben is waving there. So Ben is our industrial partnerships manager, really there to try and bridge those collaborative interactions between the work that's going on in the academic labs and commercial partners. Wendy is providing administrative support, and we're really excited that our first member of the Data Coordination Centre is starting next week or the week after, so Taya will be our initial data steward. But just to say, if people are interested in taking a lead on our Data Coordination Centre, we are recruiting. We're really looking for a data lead to drive that forwards. We're looking for RSEs and bioinformaticians, so please come and see me or drop me an email if that's something that might interest you. Final slide, it's just to acknowledge the partners who are currently involved in the initiative and to point you in the direction of our website, which we'll be developing in the next couple of weeks with additional content. And I think that is me done. So we have a great lineup of speakers today.